Awesome. So happy Monday, happy week 10. How's everyone doing? How's life? Good. Nice. Awesome. Awesome. So in terms of logistical announcements, actually, whoops, I closed out of Canvas, but let me reopen it. And then let me share my screen on Zoom. So in terms of logistical announcements, you all had Lab 9 due last night at 10 p.m. So congrats, you officially are all done with the labs. So yay, home stretch. Woo, good job, everyone. So you only have one assignment left, which is the final exam. So the final exam, just like with the midterm, you self-scheduled it. Hopefully everybody has already scheduled your final. If you haven't, you want to do that ASAP so that you get a good time. But logistics are exactly the same as the midterm, where it's at the Triton Testing Center. No notes sheet or anything like that, but they're going to give you a non-graphing scientific calculator. One thing that I want to give folks a kind of like fair warning about, and I added a clarification to the corresponding problem. So on the RNA-seq assignment, when you had to calculate log2 fold change, hint, hint, just in case this question comes up on the final, hint, sneeze, hint, cough, achoo. Whew, I don't know what was in the air right now. So just in case this question comes up on the final, hint, hint, I don't know what types of calculators they have, and I don't know if the calculators support different logarithm bases. So this problem is going to appear, if it exists on the exam, hint, sneeze, it would appear exactly in this structure. So if a calculator only supports logarithms at base 10, if you wanted to do log base 2 of a number, you could do log base 10 of that number divided by log base 10 of 2. So I would suggest practice this before the exam, just in case, hint, it shows up on the exam. You want to be comfortable doing this without like freaking out on the day of. So that's the only thing that kind of caught my eye in terms of things that might be a little bit trickier during the exam than when you were working on it on the lab. But other than that, hopefully you should be able to practice everything. We still have lab section this week, so you don't have a lab, but the lab sections are dedicated towards like if you want to practice on any of the problems that are going to be on the final, feel free to come to the lab section. That's going to be the goal of it. They're just going to be doing some like final review. If you want to work on the practice problems, again, just like with the midterm, every single question on the final is going to get pulled from these practice problems. There are no surprises. Every single question will be from here. So if you can do every single question on here, you will get a perfect score on the final. And yeah, and then the last thing, I sent out an announcement last night, but we're gonna have a guest lecture on Wednesday. Usually I try to end courses with having a guest lecture just to show you like what's going on in the industry. So one of my past students, Lily Steiner, who's now at Eli Lilly, the like pharmaceutical company, is going to be giving a presentation about like the types of stuff she works on. We're still trying to see how much she can get cleared from Eli Lilly to like disclose because they're very like protective of their IP. But at the very least, she works on computational chemistry, so doing like molecular dynamics simulations. We didn't really talk about that at all in this class, so that might be an interesting topic to learn about. And then also just any questions you might have about like what does industry look like after you graduate? Like how did your degree at UCSD help you? So she was, I think, a CS bioinformatics major. So she's gone through all these courses. She's been through it that you all have been going through. So yeah, come prepared. Please try to come in person because I don't know if I can record it depending on the IP situation. Um, and yeah, that's on Wednesday. Cool. So yeah, so final exam that everyone's scheduled, hopefully, and guest lecture on Wednesday. Any questions about like course logistics? Yeah, go for it.
For the log twofold question you just mentioned, how do you know what the calculator will provide it is? Is it base 10 or?
Ooh, good question. So the question was for the log two fold change problem. If you're given the calculator, how do you know what base it is? Short answer is it doesn't matter. So basically, whatever base the calculator is, log of that base of x divided by log of that base of 2 will be log base 2 of x. So if they only have natural log, you do natural log of x divided by natural log of 2. But usually if it says log, it'll be log base 10. And if it says ln, it'll be natural log. But yeah, no matter what it is, as long as it's a single specific base, then log base whatever that base is of x divided by log base of whatever that base is of 2. Yeah, awesome. Yeah, great question. Any other questions about logistics? The final, we're almost done. Home stretch. Y'all got this. I believe in you. Okay. So it seems like there's no questions about logistics. So let me go ahead and Share my screen with the slides. So let's go to presenter view. So for this week, again, as I mentioned, I don't like introducing new testable topics during week 10. So nothing that I'm talking about in week 10 is going to show up on the final, which makes sense because you only had 9 labs and they're all done. And I promised you every question on the final is going to be from the lab. So um, this week, I'm just kind of focusing on like, you all did all of this cool stuff in Prairie Learn in the coding environment on there, but you didn't have to install any of these tools or any of that mess. The rationale was I didn't want to have you spend a bunch of time trying to work on the lab, but making zero progress because like this tool doesn't compile on like some new Mac OS that you have or something like that. So the main goal of this week is, in addition to Wednesday's guest lecture, I want to have you have all the tools that you need to be able to set these tools up on your own computer or in whatever environment you're going to be using. So today's lecture slides, they're going to be just kind of focusing on finishing up. I don't think they're going to take the full 50 minutes, but who knows? Um, but basically, I'm going to go through these slides where today's goals, I'm going to talk about how to set up your own local Linux environment. I'm then going to talk about Jupyter notebooks, which are kind of my personal recommendation for doing scientific computing if you're doing anything in either Python, Julia, or R. I'm going to talk about different cloud resources that you could use for compute. And then I'm going to end by talking about writing pipelines. So it turns out most bioinformaticians are probably not going to be building any tools from scratch. Usually, you're going to be learning how to use the tools that we've been talking about, like in this class, and if you might learn about other tools in other classes. And most of your job is probably going to be writing pipelines that run one tool, feed the output to another tool, and kind of do any like lab specific post processing. So I'm going to talk about some different options you'll have for writing pipelines. So before I jump in, any questions, concerns? Everyone's on board? Awesome. Okay, so first things first, the cloud environment on Prairie Learn is really good, but you're going to want to have your own local Linux environment set up just because after this class, you're going to lose access to the Prairie Learn environment. By the way, after this class, you're going to lose access to the Prairie Learn environment. So any code that you want to save, please do it like right now. I think I have it set to close Friday of finals week or something like that. So go through all the previous assignments and just like download all your code if you want. And don't post it publicly. So that's an academic integrity violation. So please, if you want to post it somewhere, make it in a private repo. And you can share it with like recruiters one-on-one if you want. But yeah, that's an aside. Okay, so local Linux environment. So most bioinformatics tools are written with Linux environments that are run from the command line in mind. So to be able to run these tools on your own, you're going to probably want to have access to a local Linux environment to practice using them. So my laptop is a Windows laptop, and for Windows machines, I recommend using the Windows subsystem for Linux. This is a relatively recent like feature that was added in, I think Windows 10 was the first operating system that had it. Basically, there's just you go to the Microsoft store and you can just like search for Ubuntu, and there's an app that you click install and it just installs it. So it used to be a headache to run Linux on Windows, but now you just kind of click a button and you get a virtual Linux environment. So anything that you've been seeing me do kind of in this like little Ubuntu terminal throughout the quarter, that's exactly what I've been doing. I have like a virtual Linux environment directly there. One thing that's really nice about it is that you can directly mount, like it automatically directly mounts your Windows system. So here, my C drive is like my actual Windows machine C drive. So a lot of the stuff that I do, even if it's like not research related, like let's say I'm calculating grades or something like that, I have my local environment in like kind of set up, and then I can do things like, oh, let me download the Prairie Learn grades and then migrate them to Canvas in bulk. I don't know how to do that in Windows, but I just write kind of like regular bash scripts and whatever, and I just run it from bash with my Windows environment mounted. So highly recommend. It's a nice feature. Yeah. If you're running Mac, the terminal app should probably work fine for most cases. So you can just like hit like what's the like is it command? Is that what the Mac like equivalent of the Windows button is? Command, yeah. So if you hit like command spacebar, it'll open up like a search thing. If you type in terminal, it'll come up. I think it's under applications, utilities, terminal, if I remember correctly. One thing I highly recommend is try to avoid installing any of these tools like manually, or at least like generally avoid installing command line tools in the terminal environment with super user access because you might accidentally break things. Instead, in the same way that Linux has like sudo apt-get install whatever, or a sudo yum or whatever distro you're using, there's a package manager for Mac called Brew. So Homebrew, if you go to brew.sh, they have instructions for how to set it up. super easy. You just copy this one command, run it from the terminal, and basically certain common applications that you might want to install on your Mac, this has a lot of different applications just kind of like natively installed. So instead of having to like manually compile and install every single program you ever want, a lot of stuff you might be able to just do like brew, install some program. So I highly recommend that. Another option is whatever operating system you have, you could always dual boot a Linux distribution alongside your main operating system. However, red, bold, underline, warning, warning, you might accidentally screw up your operating systems like your hard drive's partition management and like your boot like setup, you might break that. So only do this if you really, really want to be like a power user. Don't do this if you're just experimenting. Okay. So that's kind of like my overarching recommendation, but there's a million other options that you could use. Questions? Yeah, go for it.
You don't need Homebrew for Windows?
Oh, you don't need Homebrew for Windows. So, great question. So Homebrew is a package manager, and when I install Windows subsystem for Linux, like the Linux environment that you're running is like straight up Linux. So, every modern Linux distribution typically has a package manager. So for Ubuntu, you could do like sudo apt-get install and then like whatever package you're installing. So like, let's say like I'm installing htop, and it'll like install whatever application you're doing. So with the Linux terminal, with the Mac terminal, it doesn't have its own built-in package manager, which is why you would install brew. But if you're using Windows and then you do Windows subsystem for Linux, you're good to go out of the box. Yeah. Yeah, go for it.
So in general, we can just trust Homebrew for whatever package they distribute because so I tried downloading FastQC from their official website, but I found the brew install is just way easier. But is it like pretty much the thing they distribute?
Yeah, good question. The question is, can you kind of like if you're running Mac, can you blindly trust that the stuff that you install from Homebrew, I guess, are you saying by trust it, do you mean like safe or do you mean up to date? Or do you mean works or all the above?
Yeah, just installing.
Yeah, so in terms of safety, I feel like it's a pretty safe environment because it's like open source and publicly vetted. Sometimes the applications you try to install from it might be a couple versions behind the most recent. But also if you do stuff from Linux and you do like sudo apt-get install whatever, those are also usually a couple versions behind because like usually you avoid putting the most up-to-date version in like a package manager to avoid like if there's a bug that no one caught, you don't want it to break everything. So short answer is yes, you could typically trust it if it has it. There's some other considerations to have where sometimes if you install something from a package manager, it might not be like optimally compiled for your unique computer's architecture. So there sometimes might be performance benefits if you compile something from source, but that's like very marginal benefit if that. So I usually recommend if it's on brew, just install it from there. And similarly, if it's on apt-get, with bioinformatics tools, like the apt-get package manager in Ubuntu is typically pretty far back in time, like it's pretty out of date. So bioinformatics applications I usually recommend install from source, but more kind of fundamental things like parallel or like gzip or zip or stuff like that, I would just install using the package manager. Yeah, great question. Awesome. Yeah, any other questions about this? Anything folks have like run into while you're trying to tinker on your own machine? All good? Awesome. Okay. So yeah, so that I would just highly recommend at some point. It's good to have a local Linux environment just to play around with. In the real world, usually most bioinformatics stuff is like too large for your personal computer anyways. So in practice, usually what you're doing is like remote connecting to some server and then doing stuff from like a Linux command line on some server. But it is nice to have your own local Linux environment just to like practice running scripts locally and then like deploying them on the main server. So that's usually how my development process goes. I'll write scripts on small data sets on my own computer. And then once I'm confident-ish that it's working, then I'll deploy it on the full data set on some server where it'll like blow up my computer, but it'll hopefully not blow up the server. Awesome. Okay. So the next thing I want to talk about is Jupyter Notebook. So wet lab scientists, when you're doing biology experiments or chemistry experiments, you use a lab notebook to keep track of your methods and results because you need your research to be able to be reproducible. So You write every single protocol in your lab notebook, you write the results, and then that way, 10 years from now, someone can look at your lab notebook and be like, oh, this is exactly how they did it. I can reproduce this experiment. So dry lab scientists, and by the way, the terms wet lab and dry lab just means like pipetting and like actually doing biological experiments. Dry lab is like research on a computer. But yeah, dry lab scientists should be just as meticulous about their methods and results as wet lab scientists. So one option is you could just be really, really good at documenting your code. That's always great. For me personally, I like using what are called Jupyter notebooks. So a Jupyter notebook, it's a digital notebook for keeping track of data analysis. Some fun history. The name Jupyter, it means, it stands for Julia, Python, and R. These are 3 different programming languages. So it's a notebook that you can put markdown text for like actually like rendered kind of formatted text. And you can also put code blocks that are runnable. So you could put runnable Julia, Python, and R code. And what's nice is when you save this notebook after running it, it saves your text, it saves the commands, and if there's any output results, it'll save the results directly in the notebooks. Then that way, like you could save this notebook, send it to someone, and then all of your results and methods are kind of in one place. It's also a plain text file, so you could put it in a GitHub repo and then you can version control it. So really good for reproducibility. Yeah, as I mentioned, it also supports Markdown, so you can use Markdown for text annotations, descriptions, notes, etc. For me personally, I don't actually usually use Jupyter on my own machine. I use what's called Google Colab, which is basically like how Google Drive has Docs, which is their Word equivalent, or Slides, which is their PowerPoint equivalent. They have Colab, which is their Jupyter equivalent. So you basically get a notebook environment in the cloud that kind of runs on Google's cloud compute, and they have like I think you can run it for free with like a 2-threaded machine. I forgot what the memory limits are and like what the storage limits are, but for most day-to-day stuff that you're doing, it's plenty. So I like doing that because instead of worrying about forgetting to like push my changes to some like Git repo or something like that, Google Drive is just in the cloud, so I can like access it from my laptop, from my desktop computer, wherever I am. And then also you don't have to set stuff up, which is nice. Awesome. Questions? Concerns? And I'm not going to give like a lengthy lecture on like how to set up Google Colab or Jupyter, but like you can just Google it and there's plenty of tutorials. Okay. So the next thing I'm going to talk about is what are some cloud resources that are available to you. So in addition to running things locally, there's a lot of really nice cloud resources for running stuff in some cloud compute environments. So GitHub is a great platform for version control, and then that way you can have like a distributed backup system of having version control on all of your machines that are all synchronized with each other. And then also it's stored on GitHub servers, which is nice. You all as students have access to GitHub Education, which gives you a lot of premium features for free. So definitely check that out. So for me, I personally create a different GitHub repo for every single tool that I write, and then also for every single analysis or like project that I'm starting. So like before I do any actual work, I just create a GitHub repo saying like blah, blah, blah project. And then I put all of my scripts, all of my data, everything that I can in that repo just to kind of keep it nice and version controlled. One nice feature about GitHub is after it was acquired by Microsoft, which was kind of like a nervous situation because sometimes acquisitions by these big companies kind of ruin something, or it can be good. In this case, I feel like that was a good result. Where basically Microsoft has Azure, which is their like cloud compute environment. And with GitHub repos, now that it's owned by Microsoft, you can basically click create, I think they call it code spaces, where you can basically get a cloud terminal in your GitHub repo, like in the browser. So you don't have to do Google Colab. You don't have to like set up some remote connection to some server. It like spins up a virtual machine that you can directly connect to within your browser without any headache. And it's kind of like synchronized with your GitHub repo. So a lot of times I'll do cloud development directly in there. You can like run stuff from the terminal. It's not anything beefy, but it kind of gets the job done for small tasks. As mentioned before, Google Colab is Google Drive's Jupyter Notebook environment, and behind the scenes, it's using Google's cloud platform, which is their compute, like cloud compute system. And then separately, you can kind of manually set up and configure any of these basic cloud compute services. So like Amazon has AWS, Google has Google Cloud Platform, Microsoft has Azure. One thing to warn you about though is these can be super, 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 super expensive. And if you accidentally like spin up some cloud compute resources, like maybe your lab gave you access to it and you accidentally left it running, your PI might get a bill for like $10,000 and that's not going to be happy. So just be aware, these are very easy to like screw up and accidentally like charge your boss a lot of money. For students, they have some limited free resources that are relatively safe because if you accidentally use it up, it'll just kind of like shut down your account, which is good. But yeah, just beware. Like, don't like try to set these up completely from scratch on your own, or like be very, very careful when you do. Awesome. Questions, concerns, everything all good? Okay, so in your future labs, kind of historically, bioinformatics labs usually had like a big beefy server that they were running stuff on, or like at UCSD, we have the Triton Supercomputer Center where it's like a shared compute resource. Nowadays, more and more labs are pushing towards these cloud compute resources. So if you join a bioinformatics lab at UCSD, you might want to get some practice using whatever education versions of these there are, just to kind of get a handle on like, how do I spin up a cloud environment? How do I like connect to it? How do I shut it back down? It'll save you a lot of time and it's a good kind of byline to have on your resume. Awesome. Okay. So the last section I want to talk about is writing pipelines. So as I mentioned, you're typically, each bioinformatics tool is typically written to do one very specific task. So like when we were doing the viral phylogenetics, we had a consensus sequence generator, which was input is mapped reads, output is consensus sequence. And even before that, read mapper, input is unmapped reads, output is mapped reads. Then a bunch of consensus sequences, we ran multiple sequence alignment input, a lot of unaligned reads, sequences, output is a multiple sequence alignment. Phylogenetic inference, input is multiple sequence alignment, output is unrooted tree, etc. So each tool is typically doing one kind of specific singular task. And as a result, most actual bioinformatics analyses, you're kind of chaining together multiple tools. So on the labs, we kind of isolated each individual tool where I gave you just the inputs for that one step, and you just did that single input to output. But in practice, you're probably going to have a big pipeline that's doing a bunch of steps one by one. So yeah, we call these end-to-end chains of tools pipelines or workflows. And in practice, very few people in bioinformatics are actually building new tools, and instead, you're typically just writing different pipelines that are kind of tailored towards specific projects that you're working on. So depending on like what type of sequencing experiment the lab is running, depending on like what animal it is or like what virus or whatever, there's kind of like some fine-tuned either parameter tweaks or tool choices that kind of depend on the project. So how can you actually write these pipelines? I'm going to go through a few different options and I'm going to give you the relative pros and cons of each. So the simplest thing to do, which everyone here is super, super expert at after 9 weeks of doing this over and over and over and over again, is just write a bash script. So a bash script, you just write a script. You can run it one command at a time. You can feed the output of one tool into the next tool. Another nice feature about bash scripts is that you can also use pipes to pipe data between programs to avoid unnecessary disk I/O. So hopefully if folks have taken like CSE 29, have folks, wait, is that a requirement? Are you no, it's not a requirement, but you took it. Okay. So how many people have heard of the memory hierarchy? A couple people. Okay. So this is very, very important for bioinformatics. And this was like something that I had a collaboration with Rob Knight where we like basically got a free paper out of just figuring out, hey, you guys are running stuff in a very bad way with respect to the memory hierarchy. Let's just change how you're running it. And then like things sped up by orders of magnitude. But basically the memory hierarchy, it's this hierarchy where at the bottom of this, you have very large, but very slow. I don't like these terms that they're using though. Let me find a different figure. So basically the memory hierarchy, the bottom levels have larger storage, but they're much slower. Versus, dang, these all suck. It's because Brave has, I like Brave as a browser, but their search engine is a little whack. Okay, there we go. Compute, yeah, this is good enough. Okay. And then click. Okay. So yeah, so the memory hierarchy, the bottom levels, they typically are much bigger, but they're much slower. And then kind of going up this hierarchy, you get smaller and smaller and smaller. but faster and faster and faster. And this is kind of representing getting closer and closer to where the compute is actually happening. So here, this says magnetic tape. You're probably never going to deal with magnetic tape anytime in your life. So you can kind of think of this as like cloud or network storage effectively. So something where it's really, really slow, but effectively limitless or like super big. On your machine, you probably have some type of optical disc. So I guess do computers still have CD-ROM drives? I don't think mine has one. But like Blu-ray, CD-ROM, stuff like that. These are like spinning discs that you use a laser to read. These are a bit faster, but quite a bit smaller. Then disk storage is even faster, but even smaller. Main memory is even faster, but even smaller. So like my laptop, I think has 8 gigs of memory. Then you get to your cache memory, which is even smaller, but super, super fast. Then you get to your CPU registers, where this you're looking at like on the order of bytes. But this is like as fast as possible. This is the memory right on the CPU. So that long rant is basically getting to the fact that let's say that I'm running some bioinformatics tool, that I'm running one tool and then feeding the output into another tool, then feeding the output into another tool, etc. So the files that I'm downloading and analyzing are on disk storage. So, oh geez, you can't really see that. Let me use green. So like the FASTQ files that I'm using are probably in disk storage. And then when I run a like read mapper, the read mapper is loading some of those reads into main memory. It's then loading some like an even smaller portion into cache memory, then to my CPU registers. Then it does some calculation. The CPU register like feeds stuff back. This feeds some stuff back into the CPU register, etc. And then the cache memory, once it's done with some stuff, it like writes it there. And then there's like a lot of exchange over here as well. And then blah, 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 keep writing. So basically, if you write a bash script that like runs tool 1, saves the outputs to a file, runs tool 2, saves the output to a file. Basically, each time that you're doing that, you're reading the data from disk, and then you're writing it back to disk. And each time you're making this jump is super, 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 super slow. So instead, in bash, you could use what are called pipes to directly feed the standard output of one tool into the standard input of the next tool. So then that way, instead of having this like bidirectional reading and writing, everything is just going through memory, so it's much faster. So what I usually do with my pipelines is I like in memory feed the data to each of the tools via pipes, and then any intermediary files that I do want to save. So like your mapped reads, even though you're going to feed them to something else, there's a lot of applications that the mapped reads are helpful for. So I might want to save that so I don't have to redo the read mapping. I would still pipe stuff, but then also there's a tool in bash called T. Maybe I'll write this because it's quite nice. TEE, which basically lets you feed stuff to standard output and also will write it to a file. So what I usually do is if I have stuff that I'm piping a bunch of stuff, but I do want to save some intermediates, I'll like pipe it to T. T will write the intermediate file to disk while also feeding the standard output, which then the next tool will write to disk. Actually, I think I have a good example. Oh, I'll show it later. So yeah, so basically this way, instead of having to basically each step of the way do an unnecessary write and read, I'm just doing a write between steps, which is still slow. If I can cut those out, that would be nice too. But at least this way, I'm not writing and then subsequently reading. Does that kind of make sense? Ish.
There's like a cool diagram in the CSE 29 website.
Ooh.
CSE29.site/lab7. And I think it's public for everyone.
Oh, nice. So someone says in CSE 29 website, there's a cool diagram. Could you post that on Piazza?
Yeah.
That might be kind of nice. Awesome. Thank you. So yeah, so in general, yeah, a lot of bioinformatics tools for this exact reason. They support writing to standard output, reading from standard input, and then if you ever want to write it to a file, it's very easy to just redirect output to a file. So the pro of using bash scripts to write your pipelines, you don't need any special dependencies. It'll just run in whatever shell that your Linux environment has. And these Unix tools that you're piping to are super, 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 super fast. So like in week 1, we did a lot of stuff with like word count and sort and grep and whatever. These Unix tools are really fast. It's very unlikely that you'll be able to write anything remotely as fast as these, because these are like 50 years of iteration that have been going on them. The con is it could be difficult to write more complex logic using just Unix commands. So for some of the labs, you probably found that like hey, doing this with a Python script is a lot more like developer friendly because I have access to like more complicated functions. I can do string manipulations easily. It's easy for me to store variables. You could probably do all that stuff using bash and like using pure Unix commands, but I'm not that good at Unix and I find myself to be like a decent programmer. Um, so yeah, sometimes if you're trying to do more complicated things than just like cut, grep, whatever, it might be kind of hard to do in Unix. And then the other problem is, let's say that you're running your pipeline and then it crashes. So like on, was it Saturday? Was that when the power outage was all over campus? So yeah, like let's say that you're running a bioinformatics tool and then boom, power outage on campus, and then now your tool's crashed. It would suck if you have to rerun the whole thing from scratch. And in Bash, it's kind of hard to write pipelines that are able to just kind of automatically pick up where they left off if it crashes or if you have to rerun it for some reason. So usually when I write Bash scripts, I just rerun it from scratch, and that's not always ideal. Okay, questions? Kind of makes sense. So that's option number one. Another option, which again, you've been doing a lot in this class, but you could actually get it a little bit more complicated, is using Python scripts. So some bioinformatics tools have direct Python bindings. So like PySam is a Python package that wraps around HTSlib, which is what's behind the scenes of SAM tools. There's a tool called Mappy that I think wraps around Minimap too. So you can actually like call bioinformatics tools directly from Python packages. And then another thing that I do all the time, like this is literally the majority of code that I write. I use Python subprocess module, which is literally able to execute stuff from the command line directly in Python. So you can do like subprocess.run, and you give it a list of the space sep like if you were to run a command like space separate. Let's say I want to do like I don't know, minimap2 dash a dash xsr like I don't know, reads 1.fq, reads 2.fq, for example. So like let's say I wanted to run that Unix command, I could do something like subprocess.run minimap2 dash a dash x sr reads 1.fq, reads 2.fq, and it'll work perfectly fine. It'll actually execute it from the command line. What's nice about subprocess is you can also pipe stuff. So like if I wanted to do minimap2 and then pipe the results to SAM tools, it's a little trickier, but you could just Google like Python subprocess run pipe, and it's not too bad. And once you do it a couple times, you could just reuse your existing code examples. But yeah, you could pipe stuff, and this is usually what I do if I have something a little bit more complicated. So a pro of this is, okay, well, it's not as basic as Bash. So, it is a dependency. Like you have to have Python installed on the computer that you're running it on. But most modern environments have Python. So like it would be very surprising for you to be trying to run bioinformatics tools in an environment that doesn't have Python. And what's nice is you could do a lot more complex logic than Bash. So you can like use data structures. You could like do all sorts of crazy stuff. One con is that it could be much harder to chain tools together than in Bash. So with Bash, I could just write a pipe and then pipe a bunch of stuff together. And then with T, you could do like really complicated pipes that are going like multi-directional and all sorts of crazy shit. So it could be much harder to chain tools together. You kind of have to do things somewhat linearly. And then again, just like with Bash scripts, usually if your Python script crashes, it's kind of hard to get it to pick up right where it left off. Like both Bash and Python, I could write complex logic that like maybe after each step, I like save some temporary file that denotes what steps have completed. And then maybe next time I run it, I load that file and parse it and determine what I've done. But that's a pain in the butt. So yeah, you could do it, but it's very hard to write pipelines that can just like pick up where they left off. Okay. Questions, concerns? So yeah, Bash, super basic, but super compatible and super fast. Python, a little bit more complex, which is nice, but not as performant in terms of like piping stuff together necessarily. Okay, so the last thing I want to talk about is workflow management systems. So even more sophisticated than Python, you can use what's called a workflow management system. So there's a lot of different options. They all kind of do the same thing, so you would just kind of pick what you want to use. Some of the most popular ones that I've seen, SnakeMake is a very popular pipeline management system. There's another one that's growing in popularity called Nextflow. And what's nice about Nextflow is that in addition to the pipeline system itself, they also have this NF core like series of best practice pipelines. So instead of you having to write it yourself from scratch, you might be able to just pull one of their existing best practice pipelines and just kind of use it out of the box. So they have like an RNA-seq pipeline that like what's nice about these pipelines is as you saw with each of our labs, a lot of times there's a bunch of different tool options for each task. So the pipelines that they write, you could just kind of pull them off the shelf and you actually have the ability to pick and choose which specific tools you want to run for each step if there's any type of situation. They also have these like really pretty visuals, so I kind of like these. Makes it much easier to see like what the pipeline is actually doing. Accept. Okay. And yeah, these are usually like very well documented, and these pipelines will typically also have a paper associated with them. So it's like nice because you can kind of like, instead of slapping together something on your own, which is how a lot of bioinformatics happens, it's a little bit more reproducible. Okay, so one pro about this is these pipeline tools, they're typically able to determine, like you define the pipeline, you define each step as depending on some specific previous output, and they can kind of right off the bat, and I'll maybe re-pull up that figure. Like let's say that my pipeline kind of branches into multiple steps. The pipeline tool can determine what steps are able to be run independently of one another, aka in parallel. So a lot of these pipeline tools will automatically parallelize your individual analyses. So like let's say you're doing like read mapping that then feeds to viral consensus, but also like a variant caller. It'll automatically branch those out and run those two steps in parallel if you have the computational resources for it. So you can kind of define, I want to use this many max threads, this much memory at max, and here's my pipeline, here's the input, run, and it does all that crazy configuration on its own. Another pro is that a lot of these can kind of just out of the box be deployed on cloud compute systems. So I can run it on my local machine to test it out to make sure my pipeline works. And then you can just like sign in with your AWS or Google credentials or whatever, and it just works. You don't have to do anything fancy. A con is that now we're getting more and more niche. So like your environment that you're trying to run it on might not have SnakeMake or Nextflow or whatever built in. So you might have to ask your system administrator, like, hey, can you please install this for me? I want to use these pipelines. So yeah. So these, I personally, like, this is kind of a do as I say, not as I do situation where I feel like I wish I was doing more of this stuff. I still write everything in just bash scripts. So be smarter than me. Use these. These are nice to use, but it is also a little bit more of a learning curve. Awesome. And that was my last slide. So we have like 9 minutes left. So I'll pause. Any questions about anything I just rambled about? Anything that folks want me to do for the next 9 minutes? I can like compile some tools. I can show you some of these things. I guess one thing I can show you is a tool that I have that like does some crazy branching with Unix. That's of interest. Yeah. Okay. Cool. Oh, question in the chat. Let's see. Yeah, let me go ahead and pull it up. Okay, so I'm gonna go to, let me close out of this. Pull this over here. Okay. Oh, and by the way, I forgot to mention, let me actually pull that up too. So by the way, if you wanted to install all the tools that we've been using in this class, so with Prairie Learn, the VS Code environment is literally just like a virtual environment that I configured myself. So it's using a technology called Docker, which is basically like a virtual environment that's kind of got effectively an operating system like packaged in. It's kind of like a virtual machine, but not quite like a virtual machine light. So a Docker image is basically just a collection of a bunch of tools that have been installed on some environment. So for this class, I have a public Docker file, which is what I use to install all the tools. Maybe I'll post a link in the chat, and then folks that are in class, you can just watch the recording if you want the link. Let me post this in the chat. Or if you go to github.com/Nima Docker, you can find it there. Oh, geez. I didn't want to do that. Delete. Yeah, so github.com/Nima Docker. I just have a bunch of Docker environments, but if you look up my like CSE 185 ones, so this one's the grading environment. So this is a command line environment without the VS Code stuff. So actually, this is probably better for you. And kind of like step by step shows how I installed the dependencies, installed the packages, how I compiled all those bioinformatics tools from source. So yeah, that might be helpful. Okay, now let me go ahead and show you the other thing I was talking about. So this is for an upcoming project with a CDC contract that I have that so far hasn't gotten cut yet. So fingers crossed it doesn't. Who knows? So throughout the COVID pandemic, I built the pipeline that we were using to sequence basically all the COVID samples that tested positive. And then for this new contract, we want to see, can we just sequence everything? So like, let's say there's a bunch of viruses of interest that we're using amplicons targeting those specific viruses. I kind of mentioned amplicon sequencing way back in week 4. So targeted sequencing of a wide range of pathogens. Can we run a bioinformatics pipeline that reconstructs all the sequences from a given sample altogether? So let's say I do wastewater sequencing, and let's say like someone has influenza, someone else has Mpox, and someone else has like SARS-CoV-2. Can I sequence all of the viruses in this sample and kind of process them together? So github.com/NimaSD/multivirus consensus, or if you just Google, or if you like search multivirus consensus in my GitHub account, you'll be able to find it. I'm going to go ahead and open a terminal and show you how this tool works. It's like really kind of like hacked together, but it works surprisingly decently, which is always nice. So let me go ahead and clone it. Okay, cloning. Okay, so in the repo, I have some example usage. So for example, let me go ahead and pull this over here. So in the repo, in the README, I have like example usage of like how to run it on the example data set that I include in here. So let me go ahead and try running that. I just literally copied it. I pasted it. Hopefully everything's working correctly and it's done. So what's happening here is this is an example data set that contains it contains an HIV-1 genome, a SARS-CoV-2 genome, and an Mpox genome. And it has simulated reads that, let me see, zcat example reads. And it has reads simulated from each of those genomes. So here you can see, let's actually pull out what are all the unique genomes. So let me search for lines that start with the @ symbol. Oh, not all of them work. So maybe start with @ and C. There we go. And then let me cut delimited by dash. Let me get the first field. So that gets just the genomes. And let me just delete that @ sign. So TR is translate, -D means delete, and I can specify a symbol. And it just deletes that symbol. So I deleted all the @ signs. Now we get a bunch of repetitive, like what are these genomes? So I can now sort, sort and then pipe to unique -C. And I can see how many reads did I get from each of these genomes. So let me example / startup fast rep for lines that start with a caret to get the headers. So I have 2730 HIV-1 reads, 8970 SARS-CoV-2 reads, and then 59160 Mpox reads in this data set. And it ran it. Basically, don't worry about what this Bloom filter means. This is a topic that we didn't talk about, but if you take CSE 100, we use Bloom filters. And basically, this Python script that I've written, it's a little bit janky in what it does, but basically, this Python script writes a bash script. So here's my bash script. it writes a bash script and then just calls that bash script. And this bash script that it writes, actually, can I just, maybe I'll do less so it wraps around. So this bash script that it writes calls minimap2, and it feeds in all of the reference genomes. So basically, I create a concatenated FASTA file that has all the reference genome sequences, and I map to all of them simultaneously. So giving it the concatenated FASTA file with all the reference genomes, giving it my reads. I output, so just a right arrow means redirect standard output to a file. 2 right arrow means redirect standard error to a file. So minimap2 outputs log messages to standard error. So I'm redirecting those to an output log file. And then here I'm piping the SAM output from minimap2 into T. And this is where things get a little bit weird, where basically this is called process substitution. So I can use a right arrow and then in parentheses put in some command. And basically T is going to try to write to a virtual file represented by, like, this process is going to create a virtual file on disk that's actually, it's called a FIFO, like a first in, first out. It's basically a queue. It's a named pipe. So it's actually a file on disk that's a pipe. So T is going to write stuff to a virtual file that is really feeding into the standard input of SAMTools view. And I'm writing the output as a BAM file. So this is kind of what I was saying. Let's see in the chat, someone asked. Oh yeah, no problem. So basically what I'm doing, actually I have my, oh, we're at time basically. Whoops. So just real quick, what this is doing is it's reading the FASTQ file from disk, and then it's mapping them in minimap2, and then it's given to T, which is, which one part of it is feeding the output. Maybe I'll do that on the bottom. So T is feeding the output to SAMTools, which is then writing the BAM file to disk. But T is also feeding it to this grep command, which is then grepping for just one of the viral genomes. So get just the reads mapped to like HIV or something like that, and then feed that to viral consensus for HIV. So maybe I'll label these. So this is read the FASTQ from disk, and then minimap2 to map them to all the genomes. SAMTools will then write a BAM, but also it'll feed this to grep for one virus that will then feed to viral consensus for that virus. And then write the outputs to file. And it actually calls grep on the other virus, runs viral consensus on that one, writes the output to a file. And like if you do it on like 100 viruses, it'll do it on all 100 simultaneously and write to file. So then this way you'll notice I'm only reading once and I'm only writing things to disk. I'm never then rereading them. So that's a little bit more kind of like efficient in terms of disk access. Okay, so that's kind of long-winded example, but hopefully if you test this out, you can then kind of pull little bits and pieces of this. Okay, so I'm 2 minutes over. Let's go ahead and call it here. Don't forget, Wednesday we have a guest lecture, so please try to show up. And yeah, come to lab section if you want to practice with the tutors and TA to study for the final. And yeah, good luck with finals, folks. Awesome.